Assalamualaikum and a very good day. So today's lecture is going to be about the proteolysis. Okay, this slide is actually prepared by Dr. Nuzu Jambari. So I will be giving the the lecture based on her notes, and this is going to be a quite general one, not very specific. So if you got any question, you can always ask me, or you can find the resources online. Okay, so you can read about the learning outcomes yourself. Alright, so a little bit of introduction. The normal microbial flora associated with food can produce a very wide range of breakdown products, as we learned in the glycolysis before, and also in the lipolysis. So, for example, if the microorganism can produce lactic acid, if it can go further downstream, it can produce other kind of acid as well, and even can produce some of the alcohol. So depending on the major product substrate attack, this microorganism can be designated as proteolytic, means that it can break the protein. Lipolytic means that it can break the lipid, or fermentative means that it can ferment the pyruvate into lactic acid and also its derivative. Because of their generally broad complement of enzymes, few types of microorganisms are exclusively proteolytic, lipolytic, or fermentative. But actually, most of the types can exhibit varying degrees of each properties, means that they are not exclusive. They can be proteolytic, lipolytic, and fermentative at the same time. And these all depending on the environmental condition and other factors. Nevertheless, many organisms are characteristically dominant in one or another of these three basic kinds of change produced in food. Okay, so means that although they can exhibit varying degree of each properties, but one factors usually be more dominant in certain particular microorganism. Okay, so if you learn about the microbiology in your first or second year, you will come across with this term, which is chemoheterotroph. So basically, chemoheterotroph is any living organism that take or produce energy from the food. Okay, so it's a very basic definition. You also learn about the autotroph, which means that they can use light and probably CO2 to produce their own food, for example, in the plant. Okay? But in this case, what we're going to learn is about the chemoheterotroph, means that in order to produce the ATP, in order to get the energy, they need to synthesize this energy from the food that we take, okay, in form of the chemical organic compound. So this is the simplified model of metabolism. So basically what happens is glucose can be broken down through a process of catabolism. Okay, remember metabolism consisted of two major processes. The first one is catabolism, breaking down, and the second one is anabolism, or building up. Okay, the process of catabolism involves several key reactions. For example, glycolysis, okay, breakdown of glucose into pyruvate, Krebs cycle, okay, the TCA cycle, electron transport chain, the shuttle of electron from one complexes to another in the mitochondria to produce the ATP. Okay, in most of the microorganism, it can involve fermentation, means that it just stop at catalysis and produce lactic acid, and also other kind of acid and also alcohols, and then eventually you will get the energy from these processes. The fermentation process will produce much less energy or less efficient than the complete oxidation of the glucose, and you will get this kind of end product after you undergo all these processes, and this end product actually can be used to build back all the macromolecules that you need. Okay, for example, if you need to produce a new protein, or you need to produce back the glucose that you broken down, or you need to produce fat, okay, you will need to use this end product, okay, the central molecules is ester CoA, to produce back all these macromolecules, and these processes all will require the ATP or energy, while catabolism usually will produce the ATP or energy, and in certain extent it can produce NADH, while this one will use NADH and FADH2. So this is the example of ATP. I think we already gone through this one before. It is basically one group of adenosine, or the type of nucleic acid that we can find in our DNA, attached to 3-phosphate group. Okay, so when you break down one phosphate group here, you will release a lot of energy that can be used to power the reaction that is happening in our body. So this is the catabolism of macromolecules. I don't think we need to go in detail or further into this one, because this is very specific. I'm just going to summarize this reaction. So basically what happens is, from carbohydrate that you eat, you will break down them into simple sugar, okay, and the simplest one can be glucose, and glucose can undergo certain reaction to break them into several precursors, for example pyruvic acid or pyruvic, and this pyruvic acid, if they have enough oxygen, they can enter into the mitochondria and form acetyl-CoA that can be used for Krebs cycle. And all these processes will form this coenzyme called as FADH2 and NADH that can keep or store the energy in the form of electron before they can be shuttled in the electron transport chain. Okay, another name for electron transport chain is chemiosmosis, because during this process, the complexes in the mitochondria will pump out the proton and cause the chemical osmosis between the outer membrane of the mitochondria and matrix of mitochondria, and this can produce energy that can be used to form more ATP from ADP. Okay, if you don't understand this one, you can refer to the YouTube, or you can come and see me. Okay, and the second one is protein. This protein can be broken down into many types of amino acid, but basically the most important one in the human body, it's not the most important one, but the 20 coded amino acids that can be found in our DNA, okay, they all can be broken down into certain carbon skeleton, okay, for example these three, and they can be fed into the metabolism, okay, metabolism backbone that will eventually make all the molecules that we can see in the sugar metabolism. So all of these reactions, they are actually related to each other, they are connected to each other. Okay, and as you can see from lipid consisted of 1 glycerol and 3 fatty acid, and upon the hydrolysis, okay, by the lipase, this lipid can be broken down into 1 glycerol and 3 fatty acid. And remember, this fatty acid can be different from each other, doesn't have to be similar. For example, the first fatty acid can be oleic acid, the second oleic acid can be palmitic acid, and so on. Okay, so they can be different, and this glycerol will eventually be fed into the backbone of the metabolism as well, involving sugars and also protein, and this fatty acid can be broken down into acetyl-CoA, okay, two carbon molecules. Okay, so for example, if this fatty acid is 16 carbon long, you will produce 8 carbon acetyl-CoA. Okay, and if the fatty acid is odd numbered, it can produce another one special molecule we call as succinyl-CoA, which is one of the intermediates in the Krebs cycle. Alright, so this is the building block of protein or amino acid. So basically the smallest building block or the smallest molecule of protein is amino acid. So if you digest them in your stomach, eventually you will produce certain amino acid depending on the food that you eat. Right. So basically the amino acid structure, it must consist of side chain, amino group, and carboxyl group. Okay, so this is the Zwitter ion neutral pH amphoteric, okay, because it means that this is the chemistry. It means that it can be both acid and base, okay, depending on the condition. So you don't really have to know in detail about this one. We just want to show you the basic building block of amino acid. Okay, so the 20 amino acid that I told you before that is coded in the DNA, they basically can be divided into several groups, and it is actually depending on the scientist or depending on how you want to look at the amino acid. So in this case, they want to look at from the three different group of non-polar. The first one means that they hate water. Second, polar means that they love the water. Okay, as you can see from here, non-polar consisted of long chain of carbon. Okay, and the third one is electrically charged. Okay, basically you can see the charge here, positive, or can be negative as well. Okay, or if you want to see from another perspective, you can group them differently. For example, maybe the number of the carbon in the carbon skeleton, or probably the presence of oxygen, or the number of the nitrogen is present in the amino acid. So basically there are many ways of grouping the amino acid, okay, together. Okay, so I think the major one is based on the three groups that I show you here, but you can group them more. Okay, for example, it can be aliphatic, okay, the red in color, or it can be essential amino acid, means that you must take them from the food, your body cannot synthesize them yourself. Okay, as you can see in this line here. Okay, it can be sulfur containing, for example, cysteine and methionine. Okay, can be can be basic, as the blue color. Okay, and you need to read the footnote as well. It says here, this chart only shows those amino acid for which the human genetic code directly codes for. Okay, only the 20 amino acid. Selenocysteine is often referred as the 21st amino acid, but it is encoded in a special manner. In some cases, distinguishing between asparagine or aspartic acid and glutamine or glutamic acid is difficult. In these cases, the code ASX, okay, for example, ASX is for this one, asparagine. Sorry, it should be ASN, but you can change them to ASX, or glutamine, okay, should be GLN, but you can convert them to GLX. Okay, I don't think you need to know in detail for this one. But basically, aspartic acid, they have a very similar carbon backbone with asparagine, only with the extra one nitrogen. The same for the glutamic acid as well. Okay, very similar to glutamine, but they store extra one nitrogen. So that's why sometimes it can be a little bit confusing for the scientists. Okay, so as you learned before in your in the chemistry, there are basically four different levels of protein structures. The first one is primary protein structure, which is just a sequence of amino acid. The second one is secondary protein structure, which is the folding of the amino acid just now. The tertiary protein structure is the three-dimensional folding pattern of a protein due to the side chain interaction. Okay, so the side chain can interact with each other and form like a 3D model of the earlier secondary protein structure that I told you before. And the last one is quaternary protein structure, which consisting of more than one amino acid chain. So many amino acid chain can come together and combine and form like a quaternary protein structure. So this is the example of the amino acid beta-lactoglobulin that you can find in the milk. So this is the secondary structure content of beta-alpha globulin. So the secondary structure means that the local folding of the polypeptide amino acid. So it consisted of alpha helix 6.8%, beta sheet 51.2%. Okay, the major kind of sequence of amino acid, and then it can be random or it can be beta turn. Okay, so this is the chemistry of the amino acid. So for the protein catabolism, we will continue in the second lecture. Thank you.